Welcome to the 5th Colonial Waterbird Program training video. This video will cover the gulls, terns, and shorebirds nest monitoring protocols. This video will include a protocol overview for the nest stages, a nesting stage guide for gulls, terns, and shorebirds with species identification, descriptions and photos, nest identification, additional information on subcolonies, and an example of incomplete and complete datasheets. The protocol you will follow for tracking GUTE species is as follows. You will 1. Visit the colony, watch for courtship, nesting, and breeding behaviors, fill out the first datasheet, which will have a total number of adults, total number of active nests and chicks in the colony, and a space to write information about habitat changes and disturbances. 3. Fill out page 2 of the datasheet by tracking the nest stages by counting adults, nests, or chicks. And finally, you will make a map and mark subcolonies that you are tracking. The species you will be looking for when filling out these datasheets are American avocets, black-necked stilts, California gulls, Forrester's terns, and the Caspian terns. There are key characteristics to look for when identifying each species. American avocets are large shorebirds. They have long legs, a long upturned bill, black and white upper parts, and a rusty or gray neck and head. Black-necked stilts are large shorebirds. They have a black face, hind neck, and back, white throat and underparts, long, thin pink legs, and a long, thin, straight black bill. Breeding adult California gulls are white-headed gulls with a medium gray back, yellow legs, and a dark eye. Adults have a yellow bill with small black ring and a red spot over the lower mandible. Forrester's terns are medium-sized terns. They have a black cap, a long and deeply forked tail, orange and relatively long legs, and wings that are white in most plumages. Caspian terns are large gull-like terns with a black cap, white body, and a large brilliant red bill with a dark tip. This photo has several different species. Sometimes the sites will consist of many species, so practicing identification or bringing a guidebook is helpful. Can you identify these species? Now I will highlight some of the species seen here. Here we have a black skimmer. It is not a primary species to survey, but they can be around the other primary species. Here we can see the orange rusty color of the face and neck and the upturned bill of an American avocet. This is a Forrester's tern. You can see the black cap and orange bill. Finally, the black-necked stilt is hiding in the back. You can see the white throat and black bill. In addition to identifying species, you will also have to identify different stages of nesting. There are several behaviors and characteristics to look out for when you're in the field. At stage 0, the birds will be at the pre-nesting stage. You will see the birds at or near the colony using displays of courtship such as rituals to attract a mate, such as dancing or building nests, mating, or roosting, which is sitting or resting in a location. Stage 1 will consist of egg laying or incubation. The adults will likely be on a nest for a long period of time or standing at a nest to turn eggs. The number of eggs will be hard to distinguish, but you will be able to get information about the number of nests. Stage 2 will consist of downy chicks, chicks that have downy feathers, very soft, small feathers. Adults will likely be at the colony feeding them or standing near them. Stage 3 will consist of larger chicks that have lost some of their down and are growing new feathers. The adults will still be nearby. At stage 4, you will see chicks that are fully feathered and are ready to leave the nest, known as fledging. For these species that you will be tracking, you might encounter very large colonies of birds. In order to help keep track of the individuals and behaviors, it is helpful to divide the colony into a subcolony. In this case, a barrier could help establish how you divide the colony. You can label one side A and the other side B. This is what the first page of the datasheet will look like. In a complete datasheet, the whole colony name is written, a full date is used, full names are recorded, military time is used for the start and end time, and why it was poor visibility is recorded. The second section will be to write a total nest count, total adult count, total young count, and total possible nest count. The complete data sheet wrote only possible nests and filled out each blank. For GUTE species, you should only ever have a total nest or a possible nest count, never both for a single species. This is the second data sheet you will fill out. This top portion describes the specific information for each stage that you can refer to when you are in the field. In this complete sheet, they named each subcolony, separated total adults from other stages. It might be helpful to imagine a line to divide the total adults from the other stages. The final portion of the data sheet has a section to include more information about where the subcolony is and describing it. It is essential that you fill this portion out the first time you designate a subcolony, but it does not need to be filled out every time you've first described each subcolony. If you add a subcolony later in the season, be sure to record it in this section. We also ask that you attach a site map with the subcolonies shown in the back of the sheet. This is a simple example of how to fill out the data sheet. For the purpose of the example, this is a western gull, but you will only be looking for California gulls. In this case, there are 2 adults, so you would fill in 2 under the total number of adults in the subcolony, and since they have a chick, you will fill out 1 for the number of chicks in stage 2, and leave the other stages blank. This concludes training video 5, the last video of the training series. Thank you for watching, and if you have feedback on how to improve our training, please contact Max Tarjan at mtarjan@sfbbo.org.